Hello all, welcome to another IB Biology video tutorial. This time we are focusing on section 1.3 of the syllabus, membrane structure. The essential idea for this particular section is that the structure of biological membranes makes them fluid and dynamic, and this relates to their function. As is often the case in IB biology, we're seeing the central premise of structure equals function and vice versa. So this is something to focus on again. But why are we talking about membranes? As you know, both prokaryotic and eukaryotic cells have a plasma membrane surrounding the cell. We have also seen that eukaryotic cells, unlike prokaryotic, also have internal membranes which help to produce the many different organelles. These images taken from your textbook help to demonstrate this. So we can see a prokaryotic cell here with a cell wall, but also the plasma membrane. Here is a eukaryotic animal cell, and we can also see the plasma membrane surrounding and enclosing the cytoplasm. And here's a couple of organelles. So we can see the outer and inner membranes that make up the mitochondria. And here is the nuclear membrane and also the rough ER surrounding the nuclear membrane, also made up of membranes. These membranes allow prokaryotes to separate their internal space from the external environment. They also allow eukaryotes to separate the different areas within the cell, and this is known as compartmentalization. We'll talk about this more soon. It also allows for a wide variety of chemical reactions to take place, either within the spaces created by the membranes or in or on the membranes themselves. This is the case we'll see with cellular respiration, in particular in mitochondria. We really want to focus on membrane structure today. It's a really interesting history, the development of the cell membrane model, and it provides a really good example of the importance of models in science, and we'll be talking more about this in class. We'll be looking at the change from the Davson-Danieli model to the Singer-Nicholson model, and particularly the evidence that led to this change. The current agreed model for membrane structure is the fluid mosaic model. All cellular membranes, whether they are in a prokaryote or eukaryote, or the outer plasma membrane or internal organelle membranes, have the same basic structure. You will need to be able to draw this model, this fluid mosaic model, and also outline the structure and function of each of the components. So that's what we're working towards. And here is the model. This is from your textbook, but I've removed all the labels, so just take a moment to consider this. You will not have to draw a three-dimensional structure. I'll give you a more two-dimensional version that you may draw. Okay, our first understanding is this. Phospholipids form bilayers in water due to the amphipathic properties of phospholipid molecules. The backbone of the fluid mosaic model is a layer of what's called phospholipids. So we'll look at the structure of these in detail. Phospholipids are composed of a 3-carbon glycerol head with one of these carbons attached to a polar organic alcohol and the other 2 carbons attached to non-polar fatty acids, which we call the tail. We can see that here, and again this is taken from your textbook. The alcohol and 2 fatty acids attached to this 3-carbon glycerol molecule. We call the head hydrophilic. It is water-soluble and polar, and therefore orientates itself towards water. The tail is hydrophobic. It is not water-soluble, and it is non-polar. And here we can see a representation of that. The glycerol and organic alcohol creating this polar hydrophilic region, and the 2 fatty acids creating a non-polar hydrophobic region. And we'll see this is very important for the membrane structure and also its function. The amphipathic relates to this nature. So molecules with hydrophobic or hydrophilic parts are known as amphipathic molecules, just like the phospholipid. Okay, so we can see here a small section of the basic structure of the membrane. So we have these hydrophilic alcoholic heads facing outwards, creating, orientating themselves towards the water molecules on the outside and also inside of the cell. This creates this hydrophobic region where the fatty acid tails are kind of pointing towards each other. And this effectively creates a barrier between the water environments on the outside and inside of the cell, or on the outside and inside of an organelle. This is another diagram taken from your study guide. I think this is a better diagram with which to practice our drawing. It's two-dimensional, and this is fine for the IB. So what you can see here is the phospholipid bilayer, bi meaning two, so two layers of phospholipids. Here is our first layer, and here is our second layer. So that's the first thing you need to be able to draw, a small section of these showing the phospholipids oriented as such. We can see here also our hydrophilic phosphate head and our hydrophobic hydrocarbon tail. So that is the start or the basic structure that makes up our membranes. And remember, this is any membrane in a cell, whether that membrane be in the endoplasmic reticulum, within the mitochondria, or the plasma membrane itself, whether it's in a eukaryote or a prokaryote. Okay, so what we see here is we're starting to approach a cell shape. So we've got our phospholipid bilayer, we've got our central area of a watery aqueous environment separated from the external aqueous environment. The hydrophilic heads face outwards and make contact with the water. The hydrophobic and the hydrophobic tails face inwards away from the water, creating this barrier. The barrier between the outside and the inside of the cell, or within the organelle, is therefore being created. This is known as compartmentalization. It's really important to separate out these environments so that cellular or chemical reactions can occur. Now we need to consider the structure and function of the other cell membrane components. First of all, there are a lot of different proteins involved in the membrane structure. So this understanding is membrane proteins are diverse in terms of structure, position in the membrane, and function. So again, we go back to our basic diagram here. The first protein we'll look at is the glycoproteins. Glycoproteins contain a protein molecule within the membrane itself with a sugar unit attached to the outer surface. These are really important for cell recognition, cell-to-cell communication, and also in immunity. There are also these integral proteins. Now these are proteins which are embedded in the membrane itself. So we can see here the integral proteins and also this pump or channel proteins. These are really important for a variety of reasons. For example, hormones might bind to these integral proteins. They may also act as enzymes. They might be important for cell adhesion. And they can also help to control the transport of molecules, as we'll see. In active transport, they are used as pumps, and in passive transport, they are used as channels. More on this in the next topic. There's also peripheral proteins, which are more, they do not transfer right through the membrane. They're sort of stuck on one side or on top of the membrane surface. So these are proteins that attach to the surface of the membrane. A classic example might be those involved in the electron transport chain in cellular respiration, which we'll look at in a lot of detail soon. So as you can see, a wide variety of membrane proteins. They're really diverse in terms of their structure, position in the membrane, and also their function. Integral proteins, by the way, are also amphipathic. So they have a hydrophilic region on the outside and a hydrophobic region on the inside. This allows them to pass right through the membrane. Proteins allow for a great diversity in membranes. So even though the basic structure is the same, there's a wide variety of different membranes with slightly different compositions. And it also leads to this term mosaic. So a mosaic, for example, is an ancient Roman mosaic where you have these small pieces of ceramic all put together to make a particular shape. So if you can imagine looking down on the surface of a cell membrane, it might look something like this, with proteins embedded into these phospholipid layers. Okay, another important component of the cell membrane is cholesterol. And this is a component of animal cell membranes. So back to our picture, we can see cholesterol over here. And most of the cholesterol molecule is hydrophobic. So it exists basically within, or quite deep down within the phospholipid layer. But it does contain one hydrophilic end. It therefore fits between the phospholipids in the membrane. So here is quite difficult to see on that first picture, so I've given you an enlarged version here. We can see that you need to draw these embedded within the phospholipid layers. Membranes must be fluid to function properly. And cholesterol helps to control this fluidity in animal cells at a wide range of temperatures. Plants do not use cholesterol for this. They use a combination of saturated and unsaturated fatty acids to control their fluidity. Okay, cholesterol in mammalian membranes reduces the membrane fluidity and permeability to some solutes. So remember, solutes are those dissolved substances in an aqueous solution. Cholesterol reduces the fluidity of membranes in mammals by restricting the movement of the phospholipids. So you can see here, this is a diagram taken from an outside source, that without cholesterol in the cold, the membranes might become too rigid, which means they are not very fluid or flexible, and they might actually break, obviously affecting the functioning of the cell. If it is too hot and you don't have the cholesterols, it becomes too fluid, too flexible, and it simply won't hold its shape. So cholesterol is very important for the functioning of the cell. It also reduces the permeability of the membrane to the hydrophilic molecules, such as sodium ions and hydrogen ions. So this means that it reduces the ability for these ions to pass through the membrane into or out of the cell. And this is important, as we'll see in the next topic when we talk about transport and membrane transport. Animal cells need to maintain a balance in the concentration of these ions inside and outside the cell for various reasons, which we will talk about in the near future. So, membrane cholesterol helps to control the diffusion, the transport of such molecules. And again, more on this in section 1.4. Now, I won't speak too much about the chemistry involved here, but just a little bit of a reminder, we are talking about ions. So if we look at an ions of water, for example, this diagram represents the fact that if you have an ion, a hydrogen ion molecule, what this has meant is basically it has donated an electron, a negative electron, and we call it H+ because therefore it has become more positive than it would have been with that electron. If we look at the hydroxide ion, we see that it has accepted this donated electron from the hydrogen. It therefore has one extra negative electron, so it is annotated as OH negative. So a little bit of chem, just because we are starting to talk about ions, but don't be too worried about that just yet. Okay, so what I'd like you to do just before you come to class is just practice a couple of times drawing the fluid mosaic model. Please make sure to show the phospholipids with the hydrophilic head and the hydrophobic tail. Please make sure to show a variety of different proteins. Please show a glycoprotein, an integral protein, and a peripheral protein. And also show those cholesterols embedded into the phospholipid layer. And remember, you really need to come in with an understanding of what those different components of the cell membrane do and why. And that's it for now. I'll see you in class. Hope you enjoyed.